Hello, everyone. Welcome. My name is Linda Isaacson, and welcome to today's EMSL LEARN webinar, where we'll be discussing synthetic habitats for soil ecology research. If you check out in the chat, it looks like we've got alumni representation from quite a few different places. Let's see here. I did see Sun Devils in there. Columbia Basin College, yay, go Pasco. University of Florida in Gainesville, again, more Sun Devils. University of Calgary, shout out to Kenton. Good to see you here. Let's see, and I see dinosaurs, though I'm not quite sure. Roar for the dinosaur. Any others? Let's see, I'm scrolling through here. Oh, Rochester Institute of Technology and UC Davis. All right, Sharon, good to see you too. Thank you for joining us. I bet Tomas is on here too. All right, so we're so glad to have you join us today. Next month, Roxy, if you can switch the slides. Next month, we're going to be discussing going beyond the ordinary, adventures in metabolomics at EMSL. So join us to learn more about the wonderland of metabolomics capabilities that we can offer your research projects. We'll be sharing the link for you to register for the May 11th webinar in the chat. And also make sure to keep in touch with EMSL by subscribing to our email and social media channels if you haven't already. Genoa is also going to be dropping those links into the chat. It's a great way to keep up to date on our open call for proposals and other opportunities to work with us. So today, EMSL chemist Arunima Bhattacharjee and PNNL Pauling Fellow Jayde Aufrecht will be walking through EMSL's newest capabilities in synthetic habitats. And these include microfluidic devices, a rhizosphere on a chip, and bioprinted synthetic soil aggregates. You're going to learn how you can take advantage of these capabilities for your soil ecology research. And in addition, Arunima is also going to introduce the DOE-funded Fabricated Ecosystems, or EcoFABs, project, and also how you can become involved in this effort through a pilot study. There will be time at the end of today's webinar for questions. So we encourage you to post your questions in the Zoom chat, and Arunima and Jayde will answer them during the Q&A. And so with that, we will begin with Arunima.
Thanks, Linda. Welcome everyone to our webinar, where we are going to discuss synthetic habitats. These are some new capabilities which we have developed very recently at EMSL, and I would like to stress the fact that these are our attempts to mimic soil the best we can. These are complementary to the existing characterization techniques for soil research, and these are reduced complexity habitats. And the reason for creating these habitats is because soil is very complex and heterogeneous and highly dynamic, where a lot of different microbial interactions occur. There are a lot of metabolites that is released into these soil matrix, and which results in a lot of interactions between microbes through quorum sensing, different kind of molecular interactions, which can be based on these metabolites that they are releasing to the soil environment. And so the spatial heterogeneity of soil in general resulting from these different aggregates and pore spaces that exist in between these aggregates creates subpopulation of microorganisms within them. And so there is parallel evolution of these microbes, which results in this huge diversity that we constantly see in our research. And so current broad scale process measurements mask these details of interactions that occur spatially between microbial populations. And through our reduced complexity habitats, this is an attempt to understand and visualize these interactions in soil. And so just to give you an example, So microfluidics in soil is recently gaining a lot of tractions, and a lot of these microfluidic devices are out there, and but microfluidics have been used in biomedical engineering for decades. And so a really good example that I can give why we need to use this is an example where in this particular paper, we mimic the gut villi using these rod-like features that you can see. So here, basically, when we had different length of these rod-like features, the biofilm morphology of these two microorganisms, Pseudomonas aeruginosa and E. coli, changed completely. And because of the change in biofilm morphology, we saw that the outdiffusion of metabolites, for example, in this case, indole, completely changed. And because of that, co-localization of these species changed. And this is a great example because it's a very simple two-community microbial community. But in soil, that never happens. In soil, we have so many different microbes and so many different types of species and families. They're interacting with each other. There are plants and there's so many different metabolites. So imagine the complexity that exists, and which is why we need a reduced environment to study, to differentiate, to separate these from each other. And so here, for example, is a soil core where there are different hotspots of these microbial communities. And these hotspots arises within the microaggregates. And because of these hotspots, there are spatial interactions between microbes. And this is what we are trying to capture in these synthetic habitats, which are complementary to bottom-up approach in soil ecology research. And so to perform this kind of experiments, we have a diverse range of soil-like microfluidic devices at EMSL. For example, here you can see this is a soil micromodel, a top-down view of a soil micromodel with a very homogeneous post-like feature. And in between the white space is the channel where microbes will grow. We also have micromodels with heterogeneous soil aggregates. Like, for example, this feature right here is a heterogeneous soil aggregate. This one is mimicking sandy loam soil. So this not only mimics the physical properties of soil, but also mimics the porosities that exist within that, the density of the porosity, how the soil pores are packed in there. This is a little more heterogeneous. We also have EcoFAB devices, which we have created at EMSL. All of these devices are used to study fungal growth, microbial community interactions, EPS water retention, fungal-induced hydraulic lift, mineral microbe interactions, and as well as carbon capture. I will be describing some of this research that we have done at EMSL to make sure that these devices actually work and are user-ready. And apart from these devices, there is also another new capability called the RhizoChip, which is primarily for growing plants. And my colleague Jade will be talking about that after this.
All right, thanks, Arunima. I'm excited to be sharing a new EMSL platform called the Rhizosphere on a Chip, or we call it the RhizoChip for short. This is a new synthetic soil habitat available at EMSL, and we developed it in collaboration with Oak Ridge National Laboratory. So the RhizoChip is ideal for researchers who are interested in studying plant-microbe interactions, because with the RhizoChip, we can actually image real-time dynamic interactions between growing plants and their microbial communities. And so that adds a spatial component to our data that's very difficult to extract from natural soils. The overall workflow is that we can grow a plant from seed within the RhizoChip, and then we can add our microbes of interest to the root environment. And because the RhizoChip is transparent, we can actually image the spatial organization of the roots and microbes in real time using fluorescence microscopy. And so this can help us answer questions like, where do microbes colonize on the plant roots? How quickly can microbes colonize? And then what does the succession of a microbial community look like when it's cultured together with the host plant? So we also have the option to pair the RhizoChip with a few different chemical imaging capabilities to map metabolites in the RhizoChip and then correlate that data to our microscopy data to help inform the different molecular interactions that are going on between plants and their associated microbiomes. So as a synthetic soil habitat, the RhizoChip's internal structure replicates several physical characteristics of natural soils. Using the shape statistics from natural sand from a beach in Hawaii and a granular media generating algorithm, we randomly generated 500 unique two-dimensional soil grains to use in our design. And so not only do these grains have the same shape characteristics of natural sands, but they've also been oriented and packed to have similar properties of natural soil as well. So this includes a porosity of about 38%, which approximates a sandy loam type soil. And this overall design is big enough to accommodate plant roots, but still small enough to fit onto a microscope stage so that we can image the interactions. And then once we create that two-dimensional design, we then extrude at 300 microns into the third dimension to give ample space for the roots to grow through that synthetic pore space. So this is what the RhizoChip looks like in person. We use a transparent and bioinert polymer called PDMS, or polydimethylsiloxane, to create the synthetic soil structure. So if you're unfamiliar with this polymer, you can imagine it's similar to what's used to make contact lenses. And then on this image on the right, I've filled the RhizoChip with a blue food coloring just so you can more easily see that pore space that the plant roots are going to be able to access while they're growing through the chip. And so normally we would fill that pore space with a plant-based liquid media that provides nutrients to the growing roots. So growing plants in the RhizoChip is relatively straightforward. Depending on the plant that you're interested in, we can either germinate the plant from seed or start a cutting on an agar plate. And then just as the roots start to emerge, we then transfer the plant to a pipette tip that guides the growing roots into the synthetic soil pore space. And so we found that the RhizoChip can accommodate several different plant species depending on what you're interested in. So far, we've tested a few different grass species like Brachypodium and tall wheatgrass shown here. We've also tested sorghum seedlings. And then we're currently testing poplar cuttings in the RhizoChip as well. And so the Brachypodium is the plant we've worked with the most. We've been able to grow it in the RhizoChip for up to 1 month. So that's roughly a third of its generation cycle. And we found that the seedlings met several developmental milestones on time in the RhizoChip, just as they would in natural soil. Then once the roots grow into the synthetic soil, we can start to image them under an optical microscope. And so what we see is that the roots navigate the synthetic pore space very well, even in some instances squeezing through very tight pore spaces, which they don't really seem to mind because they continue to grow normally afterwards. And so we can even get high resolution images of growing single cells over time. And so we can see, in some cases, subcellular organelle movement, like the organelles shown here within a single Arabidopsis root hair. So if you have a plant with fluorescent reporters, this is something that's possible to do in the system. And then another interesting application of the RhizoChip is that we can trace the roots of seedlings that grew through the synthetic soil space, and then study how that soil affects the root system architecture and how gravitropism, or the plant's ability to grow towards gravity, will help the roots navigate this pore space. So if you're interested in this kind of root system architecture research, please reach out to me after the talk. I'd love to talk more about it. And so one of the main reasons that we included the synthetic soil structure in the RhizoChip was because we expected that this physical structure would influence mass transport properties in the pore space. So things like root exudate diffusion. And so roots release a lot of different carbon compounds in the soil, which we call root exudates. And so these compounds feed the plant's microbiome, and in turn, some of the microbes confer a benefit to the plant. And because the soil structure is fixed in our RhizoChip, which by that I mean that it doesn't change between experimental replicates, that means we can actually model what root exudate diffusion might look like in the RhizoChip. And so that's what we did here. We traced the roots of seedlings that were grown in the RhizoChip into our simulation space. And then we assigned a constant flux from the boundaries of the roots to represent root exudation. And so our simulations predicted that there would be small pore spaces in the RhizoChip close to the root's boundaries where there would be an increased concentration of labile carbon. So that's indicated in the bottom panel by the yellow and orange spots. And of course, this was not something that we saw when we simulated the root exudation in an open geometry. And so to us, that suggests that the synthetic soil structure could be responsible for limiting the transport of root exudates away from the plant root. And then that could have downstream implications for the microbes that are then consuming and transforming these carbon compounds. Okay, so there's a few different ways that we can sample the molecular environment in the RhizoChip to confirm the presence of these labile carbon hotspots and then validate if that's validate what our simulations are predicting. And so one of those ways is through using Multimass Spec Imaging here at EMSL. And so for this process, we fix the entire RhizoChip and then remove the back cover to use that as a substrate for the Multimass Spec. And we can get good co-localization of organic compounds with where the root was, which is indicated in purple in the top panel. And also using this technique, we can identify many chemical species that are present in the pore spaces around the roots using an untargeted approach. So meaning we don't need to know exactly what we're looking for, which chemical compounds we're looking for before we go looking. And so we can confirm the presence of root exudate hotspots in the RhizoChip. And from a plant microbe interaction perspective, this non-uniform distribution of labile carbon can act to stimulate microbial growth in these concentrated carbon hotspots. And so we think that the RhizoChip could be a good model system for studying rhizosphere hotspots in the soil. So if you're interested in using the RhizoChip, there are two options available. Either we can do the work at EMSL and you can specify which plants you want to use, which microbes you're interested in working with, and whether you want to pair the RhizoChips to any fluorescence imaging or other chemical imaging techniques. Or the second option is that we can actually ship the RhizoChips to you with a set of instructions, and then you can work with them at your home institution. So at this point, I'll hand the presentation back to Arunima so she can introduce two other synthetic soil habitats.
Thanks, Jade. Okay, so the next capability that I'll be talking to you about is how we can do multimodal imaging on these soil micromodels. So these different soil micromodels that we have built here at EMSL, these are built with cheap polymer material. And it's highly reproducible, high throughput way of creating these chips. And these are compatible with different chemical and optical imaging techniques. So when you think about soil, it's opaque. So if we want to image or track how microbial development occurs, the only way to do that is optical microscopy. And we need something that is not opaque. And so these systems that I'll be talking to you about uses a polymer that is transparent. Here is an example of how it looked like. And so we have tested that these epoxy-based microfluidic systems are now compatible with optical microscopy. It is compatible with multi-mass spectrometry imaging, secondary ion mass spectrometry, SIMS, XPS. We can do SEM, EDEX on these surfaces. And they are also compatible with synchrotron-based techniques such as X-ray fluorescence and XANES. And in the next few slides, I'm going to talk to you about how we can do this and how using research here at EMSL, we have shown that that this is possible. And so one thing I'd like to stress is that this is one microfluidic design, but we can create the same microfluidic capability for any kind of microfluidic device design that users might have. So if you have a research question which uses a certain type of microfluidic device design, we can make them amenable for all these kind of multimodal imaging that I just talked about. And spatial analysis, for example, using optical and chemical imaging can be done on them. We can add solid phase nutrients such as chitin, and we can make hotspots of chitin within the device. If you want that, we can add soil minerals. That's something that I'll be talking to you as an example here. And this is to mimic the soil environment of choice. And again, to stress that we cannot exactly replicate soil because soil is super complex, but we are trying to reduce the complexity and trying to understand how components, biotic components in soil interact with these abiotic components and how this great diversity occurs over time. We can also extract bulk metabolites for analysis using MPLEX for metabolomics, proteomics, lipidomics, as well as RNA and DNA analysis from these devices. So all of these are, again, complementary to current existing analysis techniques for soil research. So one of the capability that we have developed here at EMSL, which also combines the multimodal imaging that I just described, is the mineral microfluidics capability. In this, we add the solid phase minerals into the device, and they are brought into the surface using an etching technique. And so we have tested this capability on two different types of microfluidic device. One is a micromodal. These devices have existed in EMSL for a long time, and we study transport of and flow patterns in river corridors, also to look at microbial interactions. And as well as we have tested this mineral microfluidics capability on the RhizoChip that Jayde just described to you. And so you can see here how in this example, in this particular example, we are using K-feldspar, which is a common soil mineral. And you can see here like the mineral particles sticking out of the microfluidic channel surface, and so that they can interact with the microorganisms. For example, this is a schematic of like a fungi interacting with the minerals on the surface. And so this is again like a top-down view of the mineral particle attached to the surface, as well as these are edges of the pillars where the minerals are sticking out of the surface. And so what can we do using these mineral microfluidics is one great example is to look at mineral weathering. And one of the research that I did here was looking at how fungi degrade minerals. So this is an example microfluidic device, a micromodel, where what we did was had a rich carbon source at one end, the C here that you can see. And at the other hand, we had a rich carbon source. And we studied how fungal bridging occurs between these two carbon-rich nutrient sources. And so these two examples here are a chip or a micromodel with minerals embedded. And the image below is the chip without minerals. And when there is presence of minerals, fungi is able to bridge these two hotspots. And there is nothing in between except those potassium feldspars. There is no media. And so the fungi is basically bridging through air inside of these pore spaces to the other hotspot. And we can see that the hyphal density is much more when there is minerals present, as opposed to when there is no minerals embedded in the chip. And fungi grows densely within these pore spaces. So why does this happen? The next thing we did is an example of another multimodal imaging, is a time of flight secondary ion mass spectrometry, TOF-SIMS. And so using this, we observed that the fungi was extracting potassium and sodium from these minerals. Even though we used potassium feldspar, soil minerals are never completely pure unless you synthesize them in the lab. We have such example minerals as well, like hematite, which are synthesized in the lab and can be used in these systems as well. So in this case, though, that was not a synthesized mineral. It's something that was extracted from soil. And so there were other sources of nutrients, such as potassium in there. And so we looked at the extraction of these inorganic nutrients by fungi. And in this example, you can see that these are fungal hyphae. which are rich in sodium and potassium in the case where there was minerals present. Even when there was no minerals present, there is always some amount of potassium and sodium in the fungal hyphae. So, but that's much lower in concentration, relative concentrations, than when minerals were present. So the idea was that like fungi was probably trying to extract these nutrients, and that was helping it to grow between these nutrient sources, which is what often occurs in soil, where there are hotspots of these carbon. And then in between, there might be drought. There is nothing present, no nutrient present. And so the way they can get from one hotspot to the other is by extracting these minerals or accessing resources from within these minerals and other hotspots. And so the other imaging technique that we looked at is multi-mass spectrometry imaging on these microfluidic devices, where we saw spatial distribution of fungal organic acids. So one of the ways that fungi is able to extract nutrients, inorganic nutrients, is by degrading these minerals. And to do so, it exudes organic acids, small molecular organic acids. And examples of this exist in literature. From bulk soil, they have seen that fungi exude these organic acids. But in this case, we are able to spatially look at the distribution of organic acids. This is an example of when the fungi has been growing for 7 days. And in this case, the fungi has been growing for 30 days within this micromodal surface. And what we see is different organic acids. And the organic acid, the food hotspot was right here. And where we see 3 different organic acid distribution co-localizing, but the tartaric acid distribution is something which occurs away from the food source. So this is something interesting, why a fungi switches organic acid production away from the food source. These are some questions that you can ask using these microfluidic devices, because you can look at a spatial distribution of these metabolites on the surface of these devices after growth of the microbial community. Another capability that we can use for imaging these is synchrotron-based techniques like X-ray fluorescence and XANES. So again, as an example, the microfluidics, mineral microfluidics that I've been describing here, we know that fungi is now exuding these organic acids, and we saw that they are transporting potassium through the hyphae. So how is it uptaking the potassium? So to look at that, we looked at different organic acid standards, and one of them was like citric acid. And we saw that this is an image of a fungal hyphae. The red is a potassium or organic. We have named it organic potassium because we don't know what is the chemical environment of that potassium. And it exists in all fungal hyphae that we have imaged using XRF, X-ray fluorescence. But you can also see these hotspots of blue colored hotspots, which is actually potassium citrate. And so the speculation is that the potassium citrate is produced by the fungi, and it chelates potassium and uptakes it and takes it inside the hyphae. And so we have seen using XANES that these hotspots contain potassium citrate, while these red areas, which is the other areas of hyphae, contains this organic form of potassium, which is perhaps converted to store the potassium in the hyphae for future use. So this is another example of a multimodal imaging that can be done to answer different kind of questions that users might have in soil research, looking at microbial community interactions, for example. And this study was conducted in collaboration with SSRL. So these are some examples of soil microfluidics that I just described, where we can add solid phase components like nutrients or inorganic nutrients like minerals. Another capability that we have been working on here at PNNL are the soil EcoFABs. And this is under the leadership of Kirsten Hofmockel. And so these soil EcoFABs also have been created using the same polymer, so that these are also compatible for multimodal imaging that I have just described. So the goal of this study, the soil EcoFAB study, to obtain a physically structured porous media or a habitat that is appropriate for studying soil microbial interactions, specifically for microbes, and so that these are reproducible and scalable across user laboratories. And so the idea is that these soil habitats should be representative of soil, first of all. They should be flexible for diverse measurements, which it is, just as I showed you. These are, we can do spatial imaging. We can also collect all the biomass, and I'll show that example. We can collect biomass, we can do multi-omics on these, we can also do bulk multi-omics on these devices, and they should be stable for consistent measurements across different user laboratories, and as well as compatible with the strengths at PNNL, which are the techniques that I just described. And so these devices are now ready for ring trial. If any of the users who are present here, if you want to engage in these ring trial or collaborate with us in this ring trial, you can contact me or even Kirsten for taking part in this ring trial. So we have 2 different soil EcoFABs which are ready for users. It is available in 2 different porosity configurations, and porosities, what I'm talking about is the pore space between pillars. And we have 2 different, one is a very fine porosity, which is around, both of these have standardized post diameter of 100 micron. And the porosities in one of the devices, 50 micron to 100 micron, and the other is a little smaller, 20 micron to 60 micron. The height of these pillars are 70 micron to standardize them. Again, the polymer material makes it compatible with multimodal imaging, as well as you can extract biomass for doing multi-omics at EMSL. We can send these devices to you, and then you can test them, do microbial incubations and imaging. You can send it back to us. We can do imaging here as well. So as an example of what we have done here using these devices is testing microbial community interactions. And so what we saw is when it was a rough porosity or increased pore space, these communities appear to have larger aggregates. And in smaller porosities, these communities appear to have smaller aggregates. We also extracted biomass, and we looked at how much protein these biomass have in both the fine porosity and rough porosity devices, as an example of omics that can be done on these devices. So these are examples of different microfluidic devices that are available here, using which you can do spatial imaging on them, as well as you can extract biomass and do bulk metabolomics. Next, Jayde is going to talk about another capability that she has developed, bioprinting of soil aggregates.
All right, thanks, Arunima. The last capability that I'm going to share today is slightly different than the 3 other approaches that we use to create synthetic soil habitats. So this is EMSL's new bioprinting capability. Bioprinting has been used in medical and human health research to recreate soft tissues and to understand cell signaling and development, but it has not yet been widely applied to environmental research. And so we saw an opportunity here because bioprinting allows us to print microorganisms directly by simply mixing the cells with our hydrogel-based bio ink. So that means that we can organize the microbes in a 3D design to study or optimize microbial interactions. And so we can use this to recreate a specific microbial habitat or to create an engineered biofilm, for example. And so this video is just an example of a structure that we can create using 2 microbial cultures. The hydrogel-based bioink is permeable, and so we can feed the microbes living inside the structure, and many microbes can actually consume and interact with the synthetic habitat. And so we wanted to use the bioprinting to try to replicate soil aggregates. And we decided to target soil aggregates because 90% of soil bacteria are associated with soil aggregates in some way. And so these are physically bound soil particles ranging in size from about 250 microns to about 2 millimeters. And so they're really special because they have a lot of small pore spaces to provide shelter to microbes and also a high surface area for cell attachment. So this is what our bioprinted soil aggregates look like. They're roughly 2 millimeters in diameter, and they're translucent, so we can image inside of them. These are printed with a gelatin methacrylate-based hydrogel, so that also has nanofibrillated cellulose. So you can imagine that these are a similar texture as Jell-O. When we look at these bioprinted soil aggregates in a scanning electron microscope, that's when we can really see how we're mimicking the structure of a soil aggregate habitat. So here you can see there's a lot of surface area for microbes to attach, and this structure is important because it allows microbes to form their own spatial niches. Because of this, microbes that would otherwise maybe outcompete each other in a liquid culture, now they could create their own spatial niche and cohabitate within the structured environment. Along with mimicking the physical structure of a soil aggregate, we can also mimic the chemical microenvironment of natural soil aggregates by mixing in different solid phase compounds with our hydrogels before we print them. And so we've successfully added in chitin, various minerals, plant litter, and as well as microplastics. So we hope that these different nutrient inputs could lead to the formation, help us understand the formation of microbial hotspots in the soil. And of course, we can also mix in the microbes themselves with the hydrogel. We've tried printing a range of microorganisms, and they all grow well in the synthetic aggregate. We've printed gram-negative bacteria like E. coli and Pseudomonas fluorescens, shown here that are able to express fluorescent proteins. We've also tried a gram-positive bacteria, Panabacillus polymixa. And so in this case, we added a live blood stain, which can quickly diffuse into the synthetic aggregate. And then we can see how the live and dead cells are organized within the structured environment. We can also print fungal spores like the Neurospora crassa, shown here. And so the species actually grew up very quickly to overtake and consume the entire synthetic soil aggregate. So that's shown in this picture. You can see the fuzzy white hyphae covering the entire aggregate. And of course, we can also print synthetic communities of microbes. So for example, here's Pseudomonas fluorescens in green and E. coli in red. And what we found was with this two-member community has a much different structural phenotype in the synthetic soil aggregate that it exhibits in a liquid culture. And so in the synthetic soil aggregate, the Pseudomonas will grow into microcolonies, and also the average distance between E. coli and the Pseudomonas is different in the synthetic soil aggregate than it is in liquid culture. And so that indicates to us that these strains might have a different interaction in the structured environment. And so we think that these bioprinted aggregates could be a nice way to study the spatial organization of soil microbial communities. And it might even help us be able to grow up microbes that are difficult to culture with conventional methods like liquid culture, but we have not yet tested them. So aside from fluorescence imaging, we also wanted to see if we could pair these bioprinted aggregates with analytical techniques at EMSL and whether we could measure microbial activity within a single synthetic aggregate. So first, to make our habitats more high throughput, we integrated our design with a 96-well plate to print one single synthetic aggregate per well. And then we ran a fluorescence enzyme assay and a plate reader to measure the rate of chitin decomposition by microbes growing within that synthetic aggregate. And so this is the same enzyme assay that we would do for a liquid culture. What we found was that the microbes degraded chitin at a similar rate within the soil aggregate as they do in liquid culture, but on average, but we also saw more variability in the results from the synthetic aggregates. And so we expect that this variability also arises from that heterogeneous structure of the habitat itself and that mass transport of the substrate to the microbes in the structure, which might be more realistic of a natural environment. This slide summarizes all the downstream measurements that we can do once we print the soil aggregates. So as I mentioned, we can do fluorescence imaging and biogeochemical assays on single aggregates. We can also add indicators to observe microbial processes, like the pH indicator dye here showing microbial uriolysis. And for this, my favorite part of this downstream analysis is that we can integrate the synthetic aggregates with EMSL's multi-omics pipeline to get metaphenomics data on printed microbial communities. So we found that essentially we can treat the bioprinted aggregates just like we're currently treating natural soil samples. And we're actually able to extract enough biomolecules from a single synthetic aggregate for analysis. And so part of the reason is because we don't have all the other chemical complexity of natural soil that might be obscuring the relevant data from our results. And so we've demonstrated that we can extract and amplify bacterial DNA from a single 2 millimeter size synthetic aggregate. And we can also do a single step extraction called MPLEX, which was developed at PNNL to get metabolite, lipid, and protein signals from a single synthetic aggregate. And so this is going to give us high metadata resolution so we can help correlate the dynamic imaging results to the multi-omics results all within the same aggregate. And so if you're interested in using the bioprinting capability for your own research, we have a lot of flexibility. We can come up with a print design together, or you can use the synthetic soil aggregate design. You can also specify which microbes you'd like to print and what environmental conditions you're interested in. And then depending on the scientific question that you're interested in, we can also pair the bioprinting with other downstream EMSL capabilities. So to summarize today's webinar, these are 4 new synthetic soil habitats that are available to EMSL users. As Arunima said, these are not meant to replace research that uses real soil, but rather they're a more complementary technique that can build back the complexity of a natural soil environment using a bottom-up approach. So if you have a hypothesis or you want to test a specific parameter to learn what its effect is on ecological interactions in the soil, we think these synthetic habitats could be a great addition to your research. And also, as an EMSL user, you'd have access to all the other great analytical capabilities at EMSL, and many of which we've shown that you can pair with these habitats. So if you have a specific project in mind, or maybe you just want to learn more, please feel free to contact Arunima or myself. Here are our emails, and then otherwise at this point, we'd like to open it up for questions to the audience, and thank you all very much.
All right. Wow. Arunima, Jayde, we have a ton of questions for you. It's been spectacular on the chat, all the questions that are coming through. So I hope that we can get through all of them because at this moment, there's about 17 questions. All right, we're gonna jump right into it. And I'm going to say the name of the person who asked the question, just in case maybe the question got garbled coming out of my mouth. And if the person who asked the question wants to clarify in the chat, if I've misinterpreted their question, okay? So for those of you who have asked a question and it didn't quite come out the way you were wanting it, let us know in the chat. So I'm going to start with Jenny Roca asked, and this is for Arunima, the charge differences among mineral types. So this is for your very first presentation. I think it was in the heat of the moment as you were talking. So Jenny, if you want to clarify a little bit more, it was regarding the synthetic soil. So are there charge differences among mineral types in the synthetic soil?
We have not explored that a lot, but definitely there is, because we can see that the surface of the devices change a little bit while growing microorganisms. And that is reflected upon addition of the minerals. And once we bring those minerals to the surface, we can separate different types of minerals. If you want to study one type of mineral versus another type, we can also mix minerals together. For example, I have done these studies with kaolinite, which is a clay mineral, And that's very prevalent in soil, which also contains other impurities like feldspars and hematite, etc.
All right. Also, Arunima, Richard Donovan wants to know, how are you modeling fluid flow in these microstructures?
Mostly through, we have used COMSOL in the past to do that. So the way some of these habitats are created, I think Jayde can talk more about that, but one of the sandy loam soils examples that I just showed, that's created through an algorithm. You can do tomography on a soil core and get an idea of like what is the aggregate sizes present there and like the distribution of porosity and mimic that into a microfluidic habitat.
Okay. So Jayde, to you now. Kenton Rod wants to know, are you having success scaling from the microchip experiments to the core scale?
That's a great question. That's definitely something that we're interested in trying to be able to understand, what the different scales are these ecological interactions are happening at. So we haven't actually tested, that kind of thing at a soil core scale. I think because some of the studies that we're using the synthetic habitats for is slightly different than what we would use soil core studies for. But it's yes, we definitely need to do something like that.
Kenton also had a follow-on question. Plants can move sediment grains, and I imagine the chip environment is static. Do you expect that this causes experimental artifacts?
Yeah, correct. The plants and the RhizoChip cannot move the synthetic aggregates aside in general. We have seen them grow through some very tight spaces and push the PDMS around. So we do see that the plants tend to grow normally for the most part. They meet several, like I said, several developmental milestones on time, but we do sometimes see increased root branching. There is a mechanical signals that this platform, induces in the plant. And so we're also interested in understanding what's going on there at a molecular level as well.
Mm-hmm. All right. And one more, or a couple more, Jayde. I apologize if I slaughter anybody's name on here. I'm sorry, but Amaret Bun asks, why are you selecting this species for growth in the RhizoChip? Is it based on previous experience, the characteristic of the plant, or an alternative purpose for the plant, like harvesting for biofuel?
Yeah, so the species that we've worked with are largely, priority plants for DOE because they are bioenergy crops. And then the Brachypodium is a model grass species that has more compact root structure, and so that makes it more amenable to growing in the RhizoChip. So we are testing, a few different plants that we can grow in there and trying to determine how long we can keep them in there. But that's generally the reason that we've chosen the plants that we have so far.
Mm. And Richard Donovan, again, is asking, I may have missed it, but how are these systems fabricated again? They're 3D printed?
So the first three, the RhizoChip and the ones that Arunima discussed, are based on microfluidics technology, so that we create sort of a mask in the clean room, and then we can mold the polymer off of that mask and create more high throughput replicates of the same device. And then the last one that I presented is a bioprinted capability. And so that one uses a direct write hydrogel-based bioprinter.
All right. Let's see. Can you work with anaerobic microbes on your RhizoChip?
I haven't tried it. I've seen microfluidics that have used anaerobic cultures, and they use an oxygen scavenger to create sort of these little anaerobic hotspots in the soil, and that's been really interesting to read about that. I haven't tried it personally, but I imagine that we could work something out.
Yeah, and I forgot to mention, this question was from Sibel Yalcin.
So I have an addition to that. We have polymers which are non-permeable to oxygen. If we make these devices using that, some of the polymers that we are working with here, we can create an anaerobic environment inside the chip, and that could be possible.
Very cool. Okay. So it looks like, let's see, Alan Konopka, rather than simulation modeling of exudate release and movement, is it technically feasible to do via light on, light off, and metabolomics visualization over time?
I think you're asking about if we can do metabolomics over time spatially. And the answer is we can. We just published a paper in January that we demonstrated we can do this. And my collaborators at Oak Ridge National Lab are working on doing non-destructive spatial metabolomics sampling in the RhizoChip too. And so, yes, there are ways that we can try to get metabolites over time.
We also have other imaging mass spectrometry capabilities at EMSL, and these devices, the microfluidic devices that we described, they can be opened up, and then we can like extract metabolites from certain hotspots, and then that could also be spatial. The resolution depends on what technique you use or what you want to do.
Okay. And I just also wanted to give a shout out. So Ashvini, we got your message and we will be contacting you about collaborating. So just know that we got your message there. And Anna McLoon asks, how do you find a medium that is compatible for plants and microbes? Is there a flow of fluid through the RhizoChip, or is it static once it's filled or inoculated? I teach microbiology, and students have been isolating soil endospore formers, and students have struggled to figure out how to co-culture plants and our microbes so that both can grow without too much mold. And that's for you, Arunima. So how do you find a medium that's compatible?
Yeah, that's hard. We are trying to do that right now, actually, in one of the projects where we're trying to look at plant material and trying to grow fungi, for example, if it grows in there, and trying to find like a common media. There is also the problem of finding a media that is compatible with all these imaging techniques that we just described. And it's kind of trial and error. The good news is fungi grows in most media. So you want to look at from the plant side and then look at which is most compatible or has the best fungal growth.